Hi, my name is Nick Cirone, and I'm a senior major in biological sciences. My faculty mentor is Dr. Linda Yasui. The project I'm presenting is a DNA damage response in glioblastoma cells. The project overview revolves around glioblastoma, or GBM, which is a highly aggressive and recurring cancer that arises from glial and stem cells within the brain and central nervous system. Most newly diagnosed GBM patients have a median survival of 1 year. In this project, confocal and super resolution microscopy was used to examine the DNA damage response and focuses on radiation-induced DNA double strand breaks, or DSB. These were detected through gamma H2AX foci formation. These foci represent an area of phosphorylation of the histone H2AX, which is an important step in DSB signaling. These foci can be seen with fluorescence green within the nuclei. Experimental cells were exposed to 2 gray of cesium-137 decay, and the overall result from this project was that super resolution imaging showed increased foci clarity, resolution, and substructure compared to confocal. The aggressive nature of GBM has a profound effect on patients. Most patients have a 5-year survival rate of about 6.8%, while younger patients lose about 80 years of potential life. Therefore, understanding the biological mechanisms of GBM and DSBs are important. In this project, super resolution images of foci formation, structure, and resolution can indicate DSB areas, extent, and mechanisms. In figure 2b, the gamma H2AX is an important initiation of DSB sensing. In figure 2b, DSB repair can be done through homologous recombination or non-homologous hen joining, or through other repair mechanisms. Inhibition of these repair mechanisms can lead to increased cell death. And super resolution of foci formation can help understand the GBM DNA processes and help identify improved therapeutic. In this project, the following methods included the U87 cell line, where experimental cells were exposed to 2 gray of cesium-137 decay. Control cells were not exposed, but both cells were grown in 10% fetal bovine serum. In figure 3, the gamma H2AX foci staining proceeded as a primary anti-H2AX antibody, followed by a secondary FITC antibody, which stained the gamma H2AX foci green, followed by a DAPI blue stain for cell nuclei. After the staining, the images were collected through the Zeiss LSM 900 microscope using 63x oil immersion, confocal, and super resolution images of Z-Stacks 3D images and maximal intensity projections were collected by the software ZenBlue. From the methods above, figures 5 and 6 represent confocal MIP images of foci. Confocal imaging did not reveal foci substructure. Figure 6, which is irradiated, contained more foci compared to figure 5, which was unirradiated. Figure 7-8 represents super resolution MIP images of foci. Foci did contain substructure and were more detailed compared to confocal. Figure 8, which was irradiated, contained more foci compared to figure 7, which was unirradiated. Figure 9 represents another super resolution image of a U87 cell. These foci were indicated with substructure by the profiling tool, which measured intensity peaks. Each intensity peak represents a substructure within the foci. From these results, a few conclusions can be made. First is that super resolution of foci formation has shown increased foci resolution and substructure compared to confocal. Further optimization of SR imaging techniques and cell staining procedures will help foci resolution, substructure, and subsequent DSB extent and locations. Improving these foci detail can help provide additional information on lesion complexity and repair processes. In further implementation optimization of SR imaging will help improve our understanding of DSBs, repair mechanisms, and provide additional targets for treatments for GBM. I would like to thank and acknowledge research rookies for their support and resources. I would also like to thank and acknowledge Dr. Yasui for resources, training, support, and advice. Thank you for listening to my presentation.